about this study, which you may have seen in the form of headlines like this. Now, most of the stories that were talking about this study did a really good job of explaining what actually was done, but if you didn't read beyond the headline, let's talk. First of all, why did they even do this? Well, sexual chromosome disorders that affect gametogenesis, or the production of gametes, sex cells, leads to infertility, which many people experience. So they sought to develop models which would better help us study these processes and disorders, as well as potentially offer therapeutic options. Most existing models to do these studies utilize pluripotent stem cells, which you may have heard of before. But when they're trying to study oocytes, or egg cells, they're usually using X0, so only one X chromosome, or XY, trying to turn a stem cell with an XY genotype into an egg cell. This is not easy, to the point of basically being completely inefficient. So this led the researchers to conclude that to do these studies, they're going to have to remove the Y and or duplicate the X chromosome. So the question was, how do they do that? First, they kind of started in a model system, as opposed to what they would probably want to use for more therapeutic type studies. So using embryonic stem cells from male mice, they knew that after so many passages, or basically duplications of the cell, then these will kind of just spontaneously lose the Y chromosome. So, checkbox there. Next, they needed a way to duplicate the remaining X chromosome. To do this, they introduced a chemical compound that's going to inhibit one of the proteins used to kind of keep track of the cell cycle, or how our cells go through their process of duplicating the chromosome and dividing. When they did this, they observed that the X chromosome would occasionally just duplicate itself. They did confirm that other chromosomes, our non-sex chromosomes or autosomes, were not at risk of duplicating. The X chromosome just happens to be a little bit special. Now it was time to start putting these to the test. So they turned these embryonic stem cells, growing them in particular media to turn them into what's called primordial germ cells. This is the first step into gametogenesis, which, with the addition of particular other mediators, they were able to grow these cells and differentiate them into oocytes. So now the testing really begins. So the first step was to turn these embryonic stem cells into primordial germ cells, which will go down the path of becoming gametes. The team then utilized various cell mediators to turn these cells into functioning oocytes. These sex-converted primordial germ cells actually became oocytes more efficiently than either similarly treated X0 or XY primordial germ cells. Additionally, these sex-converted oocytes function similarly to standard XX oocytes in terms of their gene expression, so what genes they're using. With this method worked out, they then wanted to test it in a model that was going to be more translational and have more therapeutic and research applications, where they isolated fibroblasts from adult male mice and turned them into what we call induced pluripotent stem cells. They used the same methods they just established to delete the Y, duplicate the X, and confirm that all of the other chromosomes were intact in their normal amounts. Then, after converting these induced pluripotent stem cells into oocytes, they fertilized the egg with sperm from a regular male mouse, and they placed this fertilized embryo into a normal female mouse who was capable of sustaining a pregnancy. This gave rise to offspring with about the same frequency of success as if they had used normal female-induced pluripotent stem cells from fibroblasts turned oocytes. These offspring lived into adulthood and were themselves fertile. So this study is certainly exciting. It's given us a brand new model to study sexual chromosome disorders, as well as oocyte genesis, which can help to understand better how these processes work, and potential treatments for infertility. This study also very briefly touched on conditions involving aneuploidy, what we more commonly know as trisomy, and while the success rate and efficiency was low, they did observe that a very similar method could actually correct issues of duplicated autosomes. Now, this study is really exciting. It has introduced an entirely new model to study and research sexual chromosome disorders, which could in the future give us treatment options and a better understanding of issues leading to infertility, as well as other methods and options for in vitro fertilization. But like any study, it comes with important limitations and considerations. One of the biggest ones is the frequency efficiency. While this was overall a success, the numbers of success were very low. This would definitely have to be improved before we would see it probably used commercially. The researchers also acknowledged that their method for confirming that the autosomes, or non-sex chromosomes, weren't affected with their treatments was a little bit more primitive or basic, so certainly before this would be applied to any larger scale translational models, they would want to see more confirmation that autosomes aren't being affected all the way down to single nucleotide mutations. There's also the consideration of whether or not this could introduce or affect homozygous recessive X-linked conditions, and so this would certainly have to be introduced as a screening method for any sort of translational application. And finally, as always, this was in mice. It doesn't necessarily parlay perfectly to humans, but it is still exciting as a potential path forward to help treat or provide treatments for infertility and other chromosomal conditions. So while, yes, offspring were made from strictly male cells, it wasn't without the help of a little bit of science along the way.